Could cell transplants one day restore vision in glaucoma? A new study looks at one major roadblock. Glaucoma is a leading cause of permanent blindness. In glaucoma, retinal ganglion cells, RGCs, die off over time. These RGCs are special nerve cells in the eye that receive signals from light-detecting cells and carry them through the optic nerve to the brain. When these ganglion cells are lost, the visual signals can't reach the brain, and vision is irreversibly damaged. Unfortunately, adult eyes cannot naturally regrow these lost nerve cells, so once vision is gone, it's gone for good. Scientists have long dreamed of replacing lost RGCs by transplanting new cells into the retina. If new ganglion cells could be made to survive and connect correctly, they might restore vision in people with advanced glaucoma. A promising source of new cells is stem cells. For example, skin or blood cells from a patient can be reprogrammed into stem cells and then coaxed in the lab to become new RGCs. In fact, researchers note that developing lab-grown RGCs holds the potential to someday make possible the restoration of vision for people who have lost it. However, this goal has always faced very big challenges. Retinal ganglion cells and glaucoma. Retinal ganglion cells are essentially the final output cells of the retina. They collect and bundle up visual information from the retina's photoreceptors and interneurons, then send that information along their long axons through the optic nerve to the brain. You can think of them as the retina's wiring that plugs into the brain. In glaucoma, pressure or other damage causes these RGCs to slowly die off. A medical review explains that glaucoma is characterized by selective, progressive degeneration of the retinal ganglion cells. In other words, these cells gradually disappear over time. Once that happens, the eye cannot send visual signals anymore, and vision is lost. Importantly, mammalian RGCs do not regenerate on their own. Because of this, current glaucoma treatments can only slow vision loss, for example, by lowering eye pressure. They cannot restore the lost RGC cells or recover vision that has already been lost. That is why researchers are pursuing cell replacement. The idea is to transplant healthy new RGCs into the retina to replace the dead ones. But as scientists explain, the retina of adults is not easily rewired, which makes this very difficult. Why replacing these cells is so hard? Transplanting RGCs into a retina and having them work properly faces many hurdles. One big obstacle is the structure of the eye itself. The innermost surface of the retina, next to the vitreous gel inside the eye, is covered by a thin layer called the inner limiting membrane, ILM. The ILM is essentially a basement membrane that separates the retina from the eye's interior. In simple terms, it is like a transparent inner lining on the surface of the retina. This membrane, while important during eye development, becomes a physical barrier in the adult eye. Experts have noted that the ILM may constitute a significant barrier to emerging ocular therapies, like gene therapy or cell transplants. In fact, a recent review explicitly points out that the ILM appears to be a significant obstacle to delivering new cells or treatments into the retina. In other words, when researchers try to inject new RGCs into the vitreous, the liquid inside the eye, the cells tend to pile up against this membrane instead of getting in. They literally get stuck on top of the retina. Beyond the ILM, there are other challenges. The retina has many layers of different cell types, and transplanted ganglion cells have to navigate to the correct layer, the ganglion cell layer, to function. Also, the environment of the adult retina can be inhibitory. Supporting cells called glia can form scars after injury, and inflammatory signals may discourage new cells from integrating. Even if new RGCs do survive in the right layer, they then face the enormous task of connecting properly. They must grow new axons that extend through the optic nerve all the way to the correct targets in the brain, and they need to make the right synapses with retinal and brain cells. As one review explains, key obstacles include promoting and guiding axon regeneration to central brain targets and achieving functional integration in the retina. All told, making cell transplantation work is like trying to rewire a very complex circuit in a fully built person, which is extremely challenging. The new study, Breaking Through the Retinal Barrier. A recent lab study took aim at the ILM problem. The research, published in 2026 in Investigative Ophthalmology and Visual Science, tried a clever new approach called inner limiting membrane photodisruption. In plain terms, the scientists used a special laser technique to punch tiny holes in the ILM, creating entry points for transplanted cells. Here's what they did. First, they prepared retina samples from large mammalian eyes, using cow eyes, and donated human retinas in the lab. They applied a safe green dye called indocyanine green to the surface of the retina, which coated the ILM. Then they shone ultra-short pulses of laser light at the dyed area. This combination created microscopic vapor nanobubbles at the membrane. Imagine many tiny bubbles rapidly forming and popping right at the ILM. When these bubbles collapsed, they produced very local punching actions on the membrane, opening up minuscule holes or pores in the ILM. In more relatable terms, the researchers basically used light and a harmless dye to blow microscopic bubbles that popped holes in the retina's inner lining. Think of it like gently puncturing a thin plastic sheet covering the retina, using laser pulses. These holes let cells or molecules get through the membrane where they normally could not pass. Once the holes were made, the team placed lab-grown retinal ganglion cells, differentiated from stem cells, on top of the ILM. They then observed how these cells behaved over a week in culture. They compared two conditions: retinas with the ILM left intact and retinas in which the ILM had been perforated by the laser method. The results were promising. In treated samples, the photodisruption clearly created pores in the ILM layer. This allowed the transplanted RGCs to move below the membrane into the retina more easily. Quantitatively, the study found that more transplanted cells survived and spread out on the retina when the ILMM was opened up. The donor RGCs also grew more of their characteristic extensions, neurites, deeper into the retinal tissue. In fact, the authors reported that ILM photodisruption was highly effective at enabling donor cells to integrate. A quote from the study's results says both the enzyme method and the laser holes significantly promoted donor RGC survival, enhanced cell spreading, and resulted in more neurites that extended deeper into the retina. But importantly, the enzyme collagenase had actually no effect on the human ILM, whereas the laser method did. In short, the laser punctures overcame the membrane barrier where other methods failed. What inner limiting membrane photodisruption means. To recap in simple language, inner limiting membrane photodisruption is a new technique where doctors or researchers deposit a photosensitive dye on the retina and then use short focused laser pulses to create tiny holes in the ILM. Because the dye absorbs the laser energy and forms microscopic bubbles that burst, it disrupts the membrane. It is called photodisruption because it uses light, photo, to disrupt the ILM. The study shows that this process can be very precise and local. It doesn't tear the whole retina, just makes patterned openings where needed. In effect, the procedure is like laying a very fine net on the retina and carefully poking holes through it with laser-guided bubbles. The authors confirmed that the rest of the retina's layers look normal under the microscope after the treatment, indicating that the method creates openings without widespread damage. What problem this method may help solve? This laser hole punching directly addresses a key hurdle in RGC transplantation. As noted, intact ILM normally keeps injected or transplanted cells from getting inside the retina. By creating controlled openings, more transplanted cells can migrate into the correct retinal layer. In the study, this resulted in many more cells actually taking residence in the retina instead of languishing on the surface. Why does this matter? If scientists can reliably deliver new RGCs into the retina, it brings the cell replacement approach closer to reality. Overcoming the ILM barrier means that other steps, like cell survival and connection, become more feasible. The study authors conclude that their technique can overcome a key barrier in RGC replacement therapy. In other words, one major roadblock to cell therapy has been removed. This can accelerate future research by allowing scientists to focus on the next challenges, rather than worrying that every cell is stuck at the outer membrane. What it does not solve yet, it is important to be clear, this is still early-stage lab research, not a treatment for patients. The inner limiting membrane photodisruption method solves one part of a much bigger puzzle. In this study, the cells were simply kept alive for a short time in a dish with retina tissue. The researchers did not and could not show restored vision or even real neural connections in a living eye. Many critical issues remain. For example, connection to the brain. Transplanted RGCs, even if they reach the retina, still need to send their axons through the optic nerve all the way to the brain's visual centers. So far, no one has achieved this in humans. As one expert review notes, key hurdles remain, including promoting and guiding axon regeneration to central brain targets and making the cells integrate into the retina's neural circuitry. Synapse formation. The new RGCs must form proper synapses connections with the existing retinal cells, bipolar, amacrine cells, etc., and with neurons in the brain. This network rebuilding is extremely complicated. Safety and immune response. Introducing new cells into the eye could trigger immune reactions or other side effects. The study on tissue samples could not address these issues in patients. Disease environment. The retina of a glaucoma patient may be much more hostile than the healthy tissue in the lab. For instance, advanced glaucoma often involves inflammation and scarring that could still harm transplanted cells. In short, photodisruption only makes it easier for cells to enter the retina. It does not make them work like native RGCs. Until the issues of long-distance connections and functional integration are solved, we will not have a true vision-restoring therapy. As a research review emphasizes, so far no treatments have restored vision in human clinical trials for glaucoma. The ILM technique does not change that fact. It is just one step in a very long journey. Why this research matters. Even with all the caveats, this study is a significant milestone in glaucoma research. It targets a problem that scientists had identified for years. The ILM was known to block new therapies. But until now, we lacked a neat way to deal with it. By showing a successful method to breach the ILM safely, the study opens the door for many follow-up experiments. Other labs can now use this technique to test RGC transplantation in animal models or advanced lab-grown human retina, potentially making progress faster. For patients, this work represents hope on the horizon. It is one of the first demonstrations that engineering the retina's structure can improve cell delivery. As one review on stem cells in glaucoma put it, creating healthy replacement RGCs and getting them into the eye holds the potential to someday make possible the restoration of vision to people who have already lost it. The new ILM opening method addresses a practical hurdle that stood between concept and reality. Moreover, the technique itself is minimally invasive. No major surgery was needed on the retina in the lab study, and could, in principle, be refined for use in living eyes. If later studies in animals confirm that the method is safe and the cells it delivers can connect, it could be incorporated into a future treatment. Even if full vision restoration remains years away, this research matters because it changes the map. It narrows down the unknowns and shows scientists where to focus next. Why restoring vision in glaucoma is still so hard. It must be emphasized that despite this progress, restoring vision in glaucoma remains extraordinarily difficult. Think of it this way. Even if we finally get new ganglion cells into the right layer of the retina, those cells have to essentially rebuild the optic nerve. They must grow long axons through the optic nerve head, navigate all the way to appropriate brain targets, like the visual cortex, and form precise connections. This is akin to rewiring a complex cable network in an adult system. Biological guidance cues that exist during development are mostly gone in the adult eye, making it tough for axons to find their way. A scientific review highlights this challenge bluntly. Besides getting cells into the retina, key obstacles include guiding all the transplanted cells, fibers to the brain, and making them functionally integrate into the visual pathway. None of these milestones has been achieved so far in human patients. In fact, as mentioned above, the review points out that no clinical trials have yet shown vision recovery from cell transplants or gene therapy in glaucoma. Other hurdles include ensuring the health of the remaining retina to support new cells, preventing immune rejection if non-patient cells are used, and addressing any side effects of the procedure itself. For example, using lasers and dyes inside an eye would require extreme precision to avoid damaging the retina or other structures. And after transplantation, patients would need time for the new cells to grow and connect, if they connect at all. In short, the eye and brain have incredibly precise networks for vision. Replacing lost RGCs is not like replacing a burned out light bulb. It's more like rewiring a computer with broken motherboard components. This is why most experts remain cautious. The ILM study is exciting, but it is one small step in a very large journey. Conclusion. In summary, this new study provides a clever way to get around one major roadblock in glaucoma cell therapy. By creating microholes in the retina's inner limiting membrane with a laser, researchers allowed transplanted retinal ganglion cells to enter and survive in the retina. This overcomes a practical hurdle that had prevented such transplants from working in the past. However, it is still very early stage research. We are still far from having a cell transplant treatment for glaucoma patients. The transplanted cells must still grow proper nerve connections to the brain, and many safety and effectiveness questions remain unanswered. For now, people with glaucoma should continue following their doctor's advice, lowering eye pressure and protecting any remaining vision with current treatments. At the same time, this research is a hopeful sign that scientists are slowly piecing together solutions. Each new advancement, like this one, brings us a little closer to the day when lost vision might be restored, but patience is needed. As the authors of the study note, overcoming the ILM barrier may help advance vision restoration strategies, but it does not yet restore vision on its own. The work continues, and this study maps a clearer path for the next steps in that quest. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.